Radhe Radhe to all, and I hope everybody is doing perfectly fine. So, today here we are to discuss a very important topic in the chapter of neoplasia, that is hallmarks of cancer. So, हमें पता है that in my first few lectures, in very short lectures, I had told you what is basically neoplasia. That is a uncontrolled growth of cells and the tissue. Even if you remove the normal the stimuli which evoke the changes, then the cells and the tissue keeps on growing. So what are all those factors which help them to grow? Cancer hota kyun hai? Fine. So all those main 8 signature markers which are very important in evolution of cancer, why cancer happens. So these are hallmarks of cancer. So today what we'll be doing is we'll be discussing the 8 fundamental changes which are considered the cellular and the molecular hallmarks or the signature markers of malignancy, that is cancer. So to begin with, the first जो हमारा है, first point is self sufficiency in growth signals, that is oncogenes. So self sufficiency means what? कि the cells are able to proliferate itself. Why? So we know these oncogenes जो होते हैं, so these oncogenes are basically your mutated genes. Fine. So these oncogenes are your mutated genes and they cause excessive cell growth, and there is absence of normal growth promoting factors. So even if when there is no growth promoting factors, even then these cells will keep on growing. Correct? So these mutated genes, they have the capability to keep the cells growing. So this is basically the self-sufficiency of the cells in growth signals, that is to keep proliferating itself. So oncogenes hote hain with which are mutated. So there's a terminology that is proto-oncogenes kya hote hain. So these are oncogenes which are mutated. So or they are overexpressed version of your normal cellular genes. Fine. So proto-oncogenes, when they are activated, they go into oncogenes. So basically oncogenes जो अभी mutated है, mute करके रखा है उनको. Correct? So अभी वो cancer causing नहीं है, but they have the capability. Once their inhibition is removed, they will keep on growing cancer. So ये self sufficiency of the cancer cells, they in growth signal they can give. So on basis of your gene product and factors that are dysregulated, so oncogenes we can divide into these categories, that is growth factor receptor, signal transduction proteins, nuclear regulatory proteins and cell cycle regulators. Let's see with some example. तो जो growth factor है, so जो हम growth factors के बारे में बात कर रहे हैं, so जो PDGFB है, the capital PDGFB, so they code for the platelet derived growth factor associated with, we see in PDGF, we see in your astrocytoma, which is a brain tumor. Then we see GF receptor, the growth factor receptors, जैसे ER, capital ERBB. So they code for epidermal growth factor receptor, which are associated with your adenocarcinoma of lung, then carcinoma breast. Then coming to your third classification, that is signal transduction protein. So this RAS coding hota hai for your GTP binding proteins. So what they do, they are associated with your carcinoma lung, colon, bladder, kidney. Then coming to nuclear regulatory proteins like MYC associated hota hai with Burkitt lymphoma. Then cell cycle regulators like your cyclin is associated with your multiple myeloma, mantle cell lymphoma. So there are many examples of these oncogenes. So they are basically classified into these category. You have to remember कि कौन सा inhibition कहाँ हो रहा है. So growth factor receptor, signal transduction protein, nuclear regulatory proteins and cell cycle regulator. So इनके examples I have told you. Then self-sufficiency of the oncogenes के बाद आता है, you have a term that is insensitivity to growth inhibitory signals. So what happens? एक तो क्या कि self-sufficient है कि जो cancer cells है that is self-sufficient to grow. But second है कि उस वो गया oncogene का activation. और एक होता है growth inhibitor factors भी होते हैं क्योंकि our bodies की normal functioning what happens, the cells cannot keep on proliferating, so there should be a check on it, correct? So there are growth inhibitory signals that, oh cell, you stop working, you're stopped now, you're function. So the cell also die. So there are signals instilled in our body which stops the cells to grow. So those are your growth inhibitory signals, correct? So if the cells become insensitive to those signals, like someone, we keep on saying you please study, but what you do, you become insensitive. The students, what they do, they become insensitive to all the whatever the noise, like if they consider as a noise, like, yes, why our professors are said telling us to read, so they become immune, they're insensitive. You can put your ear off to those comments. So that is being insensitive. So cells, so cancer cells जो होते हैं वो भी क्या करते हैं, they become insensitive to your growth inhibitory signals. मतलब उनको फर्क ही नहीं पड़ रहा कि इनहिबिशन के लिए उनको सिग्नल दिए जा रहे हैं, they keep on proliferating. So like carcinoma arises when there is a failure of growth inhibition due to deficiency of your normal tumor suppressor gene and its product. उसको हम कहते हैं tumor suppressor gene. तो tumor suppressor gene अगर functional है मतलब कैंसर ग्रो नहीं करेगा. अगर ट्यूमर सप्रेशर जीन में इनहिबिशन हो जाती है तो कैंसर सेल प्रोलिफरेट करने लगते हैं. That is tumor cells, tumor suppressor genes होते हैं like P53, retinoblastoma. What they do? उनको लगता है कि कोई जीनोटॉक्सिक स्ट्रेस है, एनवायरमेंटल स्ट्रेस है, तो वो क्या करते हैं, ओ सेल यू डाई. सो दे गिव द सिग्नल्स टू द सेल्स टू कम डाउन, करेक्ट. सो वो डेथ सिग्नल्स प्रोवाइड करते हैं बेसिकली. सो दे आर योर नेगेटिव रेगुलेटर ऑफ द सेल जीवन है, सेल साइकिल ट्रांजिशन. सो अगर आरबी फंक्शनल है तो सेल्स जो है वो प्रोलिफरेट नहीं करेंगी, तो वो कैंसर के अगेंस्ट हो गया, करेक्ट. लेकिन अगर हमारे रेटिनोब्लास्टोमा जीन में म्यूटेशन हो जाए फिर तो इसकी कोई सुनना ही नहीं है, कोई ग्रोथ इनहिबिटरी सिग्नल्स जाएंगे नहीं. तो दिस are seen in your some malignancy like your familial associated with familial syndromes like your retinoblastoma, osteosarcoma. So this is second very important hallmark of your cancers. They become insensitive to growth inhibitory signals. Self grow कर रहा है, self sufficiency है, and inhibitory signals को नहीं सुनना उसने. Insensitive to the growth inhibitory signals. The third point is evasion of apoptosis. So what is apoptosis? This I've already posted lectures on apoptosis, the chapter of cell injury. So here what happens? So there is evasion of apoptosis. Apoptosis is cells are bound to die. So there is a programmed cell death. कि इस cell को इतने ही time तक जिंदा रहना है, correct? So that is a programmed cell death. So tumor cells, what they do, they become resistant to apoptosis. They will not follow only apoptotic signals. So apoptosis को manage करने के लिए we have two broad categories. एक होती है anti-apoptotic factors और एक होता है pro-apoptotic factors. When the cells have to die, the pro-apoptotic factors will increase, anti-apoptotic factors will decrease. So this happens in cancer. So अगर anti-apoptotic will decrease, उल्टा हो जाएगा. In cancer क्या होगा, जो cells को नहीं, they don't want to die only, cancer cells. So cancer cells, they have the limitless like capacity, they can grow to the extent. So they will suppress the anti-apoptotic protein. So activation of anti-apoptotic protein will, what it will do, apoptosis को रोक देगा. तो अगर एक्टिव हो रहा है ना एंटी एपोटोटिक, और प्रो एपोटोटिक एक्टिविटी, जो प्रो एपोटोटिक प्रोटीन्स होती हैं उनकी कंसंट्रेशन को वो कम कर देगा. सो प्रो एपोटोटिक फैक्टर्स अगर कम हो रहे हैं तो भी सेल डेथ नहीं होगी, एंटी एपोटोटिक फैक्टर्स अगर ज्यादा हो रहे हैं तो भी सेल डेथ नहीं होगी. सो दीज बोथ व्हाट दे डू इज लीड टू कंप्लीट लाइक एक्सेसिव ग्रोथ ऑफ द ट्यूमर सेल्स. सो इसका एग्जाम्पल में देखो, अगर ओवर एक्सप्रेशन होता है ना BCL2, so this BCL2 is an anti-apoptotic factor, correct? So this association has been seen in follicular lymphoma. तो अगर increased concentration of anti-apoptotic protein, that is BCL2, occurs in follicular lymphoma. Similarly BAX जो होता है ये होता है pro-apoptotic factor. अगर इसकी concentration कम होती है, क्योंकि अगर म्यूटेशन हो रहा है ट्यूमर सप्रेसर जीन का, तो वो क्या करता है, बैक्स के कंसंट्रेशन को भी कम करता है, और बैक्स अगर कम हो गया, which is a pro-apoptotic factor, so consequently what happens, your apoptosis also decreases. So apoptosis is evaded. तो कैंसर सेल्स सेल्फ सफिशिएंट तो होते ही हैं, फिर वो इनसेंसिटिव हो जाएंगे ग्रोथ इनहिबिटरी सिग्नल्स के, वो इवेड करेंगे apoptosis को, जो नॉर्मल फिनोमेना है कि इस सेल की इतनी ही लाइफ थी, उस सेल को भी उन्होंने, मतलब उस प्रॉपर्टी को भी उन्होंने इवेड कर दिया कि वी डोंट फॉलो, लाइक वी आर इमॉर्टल, करेक्ट. सो वी विल कीप ऑन ग्रोइंग. देन इंपॉर्टेंट फैक्टर होता है वॉरबर्ग इफेक्ट. यू कैन कंसिडर इज अ डाइट फॉर द कैंसर सेल्स. सो मेनली क्या होती है, वॉरबर्ग इफेक्ट होती है कि रिफाइंड वर्जन ऑफ योर सेलुलर मेटाबॉलिज्म. सो दिस आर फाउंड इन ट्यूमर सेल्स. सो ट्यूमर सेल्स के eating habit को you can say this is a diet for the tumor cells. Warburg effects. This is एरोबिक ग्लाइकोलिसिस है ये. modified cellular metabolism, and they have high levels of glucose uptake, and glucose is what it is converted to your lactose by your glycolytic pathway. So there is increased production of your ATP, there is increased cell proliferation, and so few proteins like MYC and RAS, they contribute to your Warburg effect. So cells, cancer cells का खाना है basically ये. So Warburg effect. So growth promoting metabolic alterations. This is called Warburg effect. Then fifth point is your limitless replicative potential that I told you. So ये कब होती है पता है? इसके लिए तीन आपको ना जो immortal cancer cells जो होते हैं वो तीन उनका molecular marker है. एक होता है evasion of senescence, means they don't want to die. So they will follow any pathway not to die. Then self renewal, they can regenerate. Where the stem cell normally क्या होता है? Stem cells होता है to divide into two daughter cells. Where what happens? One of the daughter cells remain as stem cell. और सिमेट्रिक डिवीजन होती है ना तो क्या होता है डॉटर सेल्स जो होते हैं दे रिमेन एज स्टेम सेल्स दिस हैपेंस ड्यूरिंग एम्ब्रायोजेनेसिस कैंसर स्टेम सेल्स जो होते हैं दे आर हाईली proliferative cells so they have an asymmetrical division what will happen one daughter cell will remain as stem cell and the other non stem cell daughter what it will go is undergo differentiation pathway they will lose their stemness and but gain one or more function so loss of function gain of function mutation कहते हैं इसको So एक third है इसका evasion of mitotic crisis. So this how cancer cell becomes immortal is by this if evasion they don't follow the law of senescence they have self renewal capacity and they have the ability to evade your mitotic crisis also. So this is basically limitless replicative potential. They will keep on they have limitless capacity to keep on growing. Then your tumor angiogenesis. This is also indirectly related to your nutrition of the cells, nutrition of the cancer cells. So these are very important for cancer cells. तो जिसलिए हम देखते हैं ना जब हम malignancy के cancer के cases देखोगे आप, like carcinoma breast you are seeing or brain tumor you are seeing, the sections which we get on histology. So जब हम brain tumor के specimens को हम cut करके देखते हैं, because if we need to provide the correct diagnosis, क्योंकि see diagnosis of malignancy is very important because you know now oncosurgeons are there, then medical oncologists are there. So then when we report as oncopathologist if you're reporting a cancer diagnosis. So giving a correct diagnosis is very important because that alters the treatment of the patient or helps the clinician or the surgeons in guiding the treatment for the patient. So you hold a very important role in making a diagnosis. So what happens? So tumor, हाँ मैं बता रही थी आपको कि जब हम brain tumor के sections देखते हैं उसमें आप देखोगे highly proliferating vessels are there. So increased vessels, that is neovascularization. New vessels are coming up in the section. जितनी vascular capacity होनी चाहिए थी sections में tumor normal brain में उससे ज़्यादा हो जाती है. So there are neovascularization. The carcinoma cells they have new blood vessels, new proliferating blood vessels. But एक important drawback क्या होता है इन new vessels का they are very leaky, so they don't have strong wall. So that adds to your metastasis. क्योंकि ये leaky होते हैं तो जब tumor cells उस neovascularized जो channel से pass करेंगे, so what will they do? Wherever the vessels are going, so there are the cells, the cancer cells are coming out, so leaking out. So there is basically that is adding to your spread of the cancer. उसके बाद why इसमें होता क्या है एंजियोजेनिक फैक्टर्स के प्रोडक्शन बढ़ जाते हैं जो प्रमोट करते हैं वैस्कुलराइजेशन को और जो फैक्टर्स डिक्रीज करते हैं एंजियोजेनेसिस को उनका लॉस हो जाता है सॉरी इनहिबिटर्स को जो एंजियोजेनेसिस को इनहिबिट करते हैं फैक्टर्स उनका लॉस सो लॉस ऑफ योर एंजियोजेनिक इनहिबिटर्स and increased production of your angiogenic factors. So both lead to neovascularization, which is a very important point in your cancer hallmark. Then seventh is your ability to invade and metastasize. That is a very characteristic point of your malignancy. So what they do, so invade the extracellular matrix and they have the capacity for vascular dissemination, tissue homing, colonization. So all these leads to your metastasis. So metastasis when we'll discuss these points in detail. The last point है जो कैंसर का हॉलमार्क होता है that is your evasion of your immune surveillance. So tumor cells जो होते हैं ना खुद में ही they are so confident और so capable so they release your tumor antigens. उसके against फिर हमारे बॉडी में काम करना होता है cells को. So tumor antigens तीन basic three हो गए आपके neo antigens हो गए they are produced as the passenger through the passenger and driver mutation Then there is overexpression or aberrantly normal cellular proteins, and some oncogenic virus भी होते हैं जो they keep on producing your these tumor antigens. So let's recapitulate what we have discussed till now. A very important topic. That is, it comes as a long question also, it comes as a short note also. See, the hallmarks of cancer. Main 8 cellular and molecular signatures I told you. First is your self-sufficiency in growth signals, that is oncogenes. Proto-oncogenes are the mutated form of oncogenes. So oncogenes जो होते हैं वो cancer producing cells हो गए. Then second हो गया खुद तो एक तो self sufficient है ही. Second आ गया insensitive, not to hear to any growth inhibitory signals. See, I'm not going to listen to any inhibition. I will keep on doing this. Okay, then you have your evasion of apoptosis. Cells which were destined to die, they are not dying now. So they have evaded the follow of the commands of your apoptotic markers. So pro-apoptotic markers are decreased, anti-apoptotic markers are increased. So that is leading to your evasion of apoptosis. Then you have your Warburg effect. So Warburg effect is related to your nutrition of the tumor cells. So इसमें होता है कि they basically the जो glucose high intake of glucose is there, glucose is converted to your lactose by the glycolytic pathway, and so that serves as a nutrition to the tumor cells. Then fifth is your limitless replicative potential. So the limitless replicative potential कैसे होता था? एक they are evading the senescence कि I will not die. Second is evasion of mitotic crisis, and third was your self-renewal capacity, which the tumor cancer cells होते हैं, they have high proliferative cells. Then sixth is your tumor angiogenesis, the neovascularization का concept है ये. New blood vessels, new proliferating blood vessels are found in the cancer tissue, and what happens is these are leaky, these walls are strong नहीं होते, so they are leaky. So what happens? They allow for metastasis. So tumor cells become metastasized also. Okay. Then the seventh is your ability to invade and metastasize. So cancer cells, we know this is a hallmark of malignancy. So they will keep on metastasizing, going to this place, that place, other organs, deep, far distant organ, nearby organs, through the lymphatic carcinoma, all those things, they will spread through the lymphatics, hematogenous or direct seeding, which we have already discussed. The last hota hai evasion of immune surveillance. Tumor cells have the capacity to generate or produce your tumor antigens also. So all these points, ये सब मिलके क्या करते हैं कि कैंसर को बोलते हैं yes cancer cells you grow, we'll support you. So these are your basic framework under which the cancer cells grow, replicate, produce the negative complications and everything. So these are your 8 hallmarks of cancer, basically the cellular and molecular hallmarks of your cancer. So thank you so much for your patient hearing. Kindly like, subscribe and share my YouTube channel Patho with MK. Thank you so much.